Now, yes.
Okay, perfect. Today we will discuss, of course, the importance of the microbiome and more specifically I will focus on human microbiomes. The other speakers will focus on other microbiomes. We will discuss mainly a question that we addressed during a European project. And the question was about the interest of specifically gut microbiome testing. So as Stephan already briefly, human microbiomes are very important, and especially the gut microbiome because it's the most diverse and the more dense microbiome that we have on the human body. So it's very important to know that humans are microbial ecosystem symbiosis. So we are really working together with the microorganism. We can find microorganisms in all the cavities in the human body. So for sure in the gut, but also in any other microbiome cavities, we have microorganisms on the skin, in the vagina, and in a lot of other organs. We have as many microorganisms in the body as human cells, so it's a ratio of 1 to 1, more or less. And regarding the genes, we can see that we have much more microbial genes on average compared to human genes, meaning that microbial cells can really bring us a lot of function that our own human cells and human genes are not able to provide us with. So we can really ask us this maybe philosophical question, are we really 100% human? Due to this importance of human microbiomes, perturbation of the human microbiomes, and especially again, human gut microbiome is now associated with a lot of diseases. You can see here some example of diseases, and I really emphasis on the fact that it's an association because for most of the disease, the causal relationship, so really the fact that the changes in the gut microbiome are the cause of the disease are still to be demonstrated. So the cause or the consequences of the changes in the microbiome is still a very large topic of discussion and research among the scientific field. So today also microbiome science has captured public interest. So it's not only, an interest for the scientists in their lab, but it's also an interest for the general public. And one of the clear example of this interest is the rise of microbiome testing and the availability of microbiome testing kits. So you can really see more and more kits like that available on the market, both in pharmacy, but also, of course, on internet. So company can sell directly these tests to consumer to evaluate the healthiness of the gut microbiome and also to provide recommendations. And you can obtain this kind of results. So I pick up some picture from internet, so you can have really an overview of what bacteria are within your gut microbiome. So you have here different names of group of bacteria that you can find in the human gut microbiome, and you can see what are your results in color compared to an average population in gray. You can have also like example of some indexes, so such as this one of dysbiosis indexes. Dysbiosis meaning the alteration of the gut microbiome, so the unhealthiness of the microbiome with from green to red. And so if you are red, it's maybe not good. And during a European project, we asked the question about the scientific validity of these tests, because microbiome science, it's still a very recent field. So we know the importance of the microbiome, I don't know, maybe for only 20 years or maybe a little bit more. And so we can really ask what is the validation, the scientific validity of these tests. And so during the human microbiome action, we answer, No, we address this question, and I will just introduce you briefly this project. If you want more information, you have here the website, so you can check the website. You will find all the report also published and the publication published during this project. So the objective of this project was to maximize the impact of European microbiome research and innovation in order to tackle epidemics of chronic diseases. And at the core of this project mission was the ambition to create coherence and harmony in the way microbiome research is and will be performed. This project was coordinated by Joël Doré from INRAE in France. The project lasted 3 years, and the 16 partners participate to this project from different European countries and with different scientific expertise. And the PRI was one of these partners, UFIC too. So it was a very diverse expertise within this the partner of this project. So during this project, we send one sample, one fecal sample from a healthy adult donor to 6 different companies, 5 companies in Europe and 1 in the US, to obtain, to mimic a real consumer experience. So to really put us in the foot of a consumer that send the samples to this kind of company, and to see what the consumer will receive as result and feedback and recommendation from the company. So we publish the results in a scientific publication. So if you want to check the result, the full results, you can have access to the publication. We have also published some fact sheet on the Human Microbiome Action websites, and the core findings of this study was unfortunately the inconsistency in the result that we obtained from 1 samples sent to different companies. So we obtain contradictory interpretation. So some companies providing us with more, I would say, a good gut health, whether the other interpret the data as poor gut health from the same data. So it's really an issue to our point of view. And then we also obtain in the report provided with this kit vague or overstated recommendation, often not supported by a robust science. The recommendations were mainly dietary recommendations, but some of them were clearly not supported by robust scientific data. So one of the key issues of the report is the over-interpretation of the tests. And so in the some interpretation and recommendation formulated in the different reports are premature and not supported by scientific data, because, as I mentioned, the field is still a very recent field. We have some data, but some of them are contradictory also in the scientific literature. So it's very important to be to interpret the data with some caution, I would say. And then also it's also very important to be aware that the association between microbiomes, as I mentioned, there is only association between microbiomes and some disease, and some mentioning association between microbiome characteristic and the risk of developing certain disease in this kind of report can be very dangerous for the consumer. And it's this kind of kits up to date is only with the intended objective to satisfy the curiosity of the consumer and never to predict any risk or to diagnose any disease. So to increase the reliability of this kind of tests that are really promising, we need to work on different aspects. First, we need to work on the standardization of analytical methods, because currently all the scientists are using different methods with different quality criteria and so on. So it's very important to work together towards the standardization. It's a normal evolution of a very recent field. So the field will mature and we will obtain a certain level of standardization. And so maybe in the future, analyzing the gut microbiota composition will be as simple as we can know, for example, assess the blood sugar level. We have also to work on on the consensus on some key scientific definitions, such as what is a healthy gut microbiome. Today, we are unable to define what is a healthy microbiome, so it's very difficult to define also what is an unhealthy gut microbiome. So we need more data and more scientific research to address this kind of question. Then we have also the key question of biomarker validation. As I mentioned, there is only association between perturbation of the gut and some diseases. And so maybe in the future, we can really move forward this field and maybe identified a gut microbial signature that can be really predictive of a certain disease or a risk factor to develop a particular disease. And then we need also clear regulatory pathways. So we need a clear message towards the consumer, towards the patient, and also towards the health professionals about the status of this kit. Are we talking about a kit to diagnose something, or are we talking about a kit just to, assess your, for your curiosity, like you can maybe wear a smartwatch only to have some data, but the doctors will never use the data to make a diagnostic of a disease. So in this publication, we propose two parallel regulatory pathways for microbiome testing kits. One should be for curiosity only, and one should be for clinical use, in vitro diagnostics, so medical device, a very strict regulatory category where these kits can really be used to assess patient and to really make health or medical claims. But on the other side, we have curiosity tests only for curiosity, so to assess what is the gut microbiota, because you want to know what are the bacteria in your gut, and this kind of kits, to our point of view, should never be associated with any health claim or medical claim, and if they propose some nutritional advice, it should be always supervised by a healthcare professional or dietitians. To conclude, I will, so what are the consequences of the lack of reliability and oversight for these kits? So first, the first point is very important, because based on the result that we obtained during this study, we had really the impression that consumers are spending a large amount of money, because these tests are very, very expensive today, to obtain for curiosity only analysis. So they know what is the composition of their microbiome, but then it's impossible to make any recommendation based on that, because it's just too early. We don't have the data needed. Companies can also have poor feedback on the services and seeing a loss of confidence from the end users. And finally, the credibility of the entire microbiome field can be affected by the lack of reproducible results, inconsistent interpretation, and conflicting results that are present in the scientific literature. So we have really to work together to improve the validation of the analytical methods we are using, and also to work on a big consensus on some scientific definition. And the role of the PRI is to help to clarify these boundaries, also the regulatory pathway, and in the specific field of microbiome testing, we will support, of course, the development of trustworthy microbiome testing to really increase also to increase on one side consumer satisfaction, but also to increase the patient's access to tests that can really help patient and healthcare professionals to improve the clinical practice and to move towards personalized medicine. Thank you very much for your attention.
Thank you very much, Céline. We already saw like lots of questions coming in related to what is a healthy microbiome, what's a dysbiosis. Maybe we can dive into those questions after bit more. I'm curious to, we'll hear your thoughts on this. But now let's head over to our next speaker, Luca Cocolin. Luca is professor of food microbiology at the University of Turin in Italy. He's one of Europe's leading experts, if I may say so, on how microbes play a role in our food, from traditional fermentation to food safety and innovation. And he published hundreds of scientific papers and is shaping food research across Europe. Luca will walk us through how microbes in our food interact with our body and affect our everyday lives and health. Luca, so nice to have you with us today. Please feel free to share your screen and take over the stage.
Thank you very much. I'm just going to be sharing. Okay, you see my screen, just to get a confirmation. Okay, great. Thank you very much. So, I mean, thank you very much again for the invitation. I'm quite happy and glad to be part of this discussion today on this important topic that sees microbes really in the center of the attention. Today is World Microbiome Day, and so we are really celebrating it the way it is supposed to be celebrated. My role today is just to give you some insights on how the food that we are actually eating every day is influencing or modulating the microorganisms that we have in our gut. And as a consequence, they can have a role also in human health. And before entering specifically into the subject, I just want to give you a very, very fast snapshot on how the diet can really influence in a specific and detailed way the strain diversity, meaning the ecosystem that we have inside of our in our gut. And I just want to show you a couple of examples on how the diet is modulating. So in this slide, you can see in this figure, every single spot represents a volunteer that was either an omnivore, a vegetarian, or a vegan. And just mapping the microbiota that they had in their gut, I mean, you can see that there is actually no clear segregation or layers of population that have been studied, meaning that independently from the diet, it seems that the microbiota that we have inside of our gut is actually not really differentiated. And this is also confirmed at the moment we are looking into the stratification with much more, I would say, precise and deep technologies like 16S RNA sequencing, because you see here that, again, we have everything that is mixed. So based on the results that we got from these two important studies, it seems as if the diet is actually not generating any kind of classification or any kind of clusterization of the different subjects. However, if we switch from ecology-based differentiation to functional-based characterization, looking into the microbiome, here you can start to see that the different diets are actually separating. And so you see here first the metagenomics, so really looking at the functions that are expressed inside of our intestine. And here you see that the omnivores actually have different functions that are expressed with comparison with vegan and with vegetarians. And even more interesting is that if we go to metaproteomics, you can see clearly the differentiation of the three groups. What does this mean? It means that based on what we feed our microorganisms from our diets, they are actually developing in a different way. And when we're looking into function, I mean, there are specific functions that are really differentiated based on the diet. And what is even more important, in my opinion, and very interesting, is that this influence goes down very, very specific to species level and to intraspecies level. This is a study that we have published some year ago focusing specifically on this Prevotella copra, that is one species that is very commonly present in our guts. And you can see here that based on the reconstruction of the genome that we are able to do from metagenomic libraries, we could really differentiate subspecies of this species based on the diet of the subjects, meaning that the different diets are not only modulating the microbiome itself, but is also modulating single strains inside of our gut. So this is something, in my opinion, that is very, very important. And if we go now back to the main concept of my contribution today, here you see really the main two roles that diet is actually having in our life. For sure, it is important to give us nutrients. So as through the digestion, we are able to uptake food components that are serving as a fuel and as a components of our body. But we don't have to forget that there is also the other part of ourselves that Céline defined as, are we 100% human? Because somehow we are feeding also the microorganisms that are present inside our gut. We know that there are portions of our foods that we are not able to digest because we don't have the specific enzymes. And so they reach the intestine, and here the microorganisms are able to consume them, are able to intake them, but they are also able to release some of the components that, as you can see in the part below, they are actually interacting with us as a host, and so they are influencing our health. And as a consequence of this kind of interchange or interlink, it's important that these two components are really well associated. And when we are thinking about microbes and diet, I think that everybody is well aware of this differentiation. The first thing that we think when we are thinking about modulation of our microbiota and our gut microbiome is about probiotics. Probiotics have been around for decades now, and so we know that there are specific strains of specific microorganisms that can really have benefits to our health. We can help probiotics by feeding them with prebiotics. So if probiotics are microorganisms, prebiotics are food components that, as you can see, are mainly found in vegetable products where there is a richness in terms of fibers, for instance, that they can help the development of probiotics. However, one aspect that I would like to focus in the rest of my speech now is that we don't have only to think about probiotics as a source of live microorganisms in our diets, because if we take into consideration the food that we are eating, there are actually foods that they contain a huge amount of microorganisms. And those foods are the ones that we are producing through fermentation. So think about cheeses, fermented sausages, think about fermented vegetables. Those are really the ones that have a very, very big number of microorganisms. And so probably unconsciously, at the moment we are consuming fermented foods, we are introducing in our body a lot of microorganisms that for sure may have an influence on our microbial composition or microbial modulation. And this is something important because fermentation at this point is really at the center of this transformation from raw materials to fermented products, because we have been used to understand fermentation as important sustainable processes through which raw materials can be transformed in products that have longer shelf life, they are more safe, but we don't have to forget about the impact that they may have on our health. And if you can see in this slide, it is very obvious how through fermentation, we are transforming our raw material in a kind of product that contains some substances that are very important for our health. Imagine that there is a lot of microbial activity, there is a lot of enzymatic activity. So somehow some people consider fermented foods as a kind of pre-digested foods that then we are consuming. But it is true that the benefit that may come from the consumption of fermented foods is not only related to the metabolites, but is also related to the presence of specific microorganisms. And so fermentation is really an important aspect. However, I really want to conclude my presentation here by pointing out a very, very important aspect that has to be highlighted in order to avoid confusion. Often, it happens that when we are looking for fermented foods, the word probiotic is appearing very, very often. We don't have to be mistaken, or no, we don't have to have confusion on the terms fermented foods and probiotics, because those two things are actually not the same unless the fermented food has been produced with a specific probiotic microorganism. And so in this opinion that the HISAP has published some years ago, you can see really when we have to define, or when we can define a product as probiotic or not probiotic, or a probiotic fermented food. And it's interesting here just to take into consideration that when we have a fermented food, you can see that is not always present as a live microorganism, because we can have cooked or processed fermented foods. Think about the bread, for instance. And also, we don't have the necessity to have a taxonomical defined strain level, while it is extremely important for probiotic and also for probiotic fermented food. So again, the take-home message here is we don't have to consider fermented foods as probiotics unless we have proven that inside of the fermented food there are probiotic microorganisms, and this is not always straightforward. And I just want to finish, because my time is off, by pointing out another aspect in this moment in which fermentation is acquiring a lot of attention, and it seems that technology, and I want to consider it as a technology that is thousands and thousands and thousands years old, has been rediscovered as one of the most innovative solutions that we can have in the food system. Let's not forget about safety. I mean, the concern that we may have when we are pushing for fermentation that is done also in contexts that are not the industrial settings, where fermentations are known, are controlled, and they are somehow kept to a certain level of safety, may create issues for human health. So since we have been used in the past to start to use thermometers to understand the temperature of our meat when we are cooking it, in order to make sure that the temperature in the core of the product is reaching a safe value that is eliminating all the microbiological risks, well, I think that we should also push for another kind of application, like, for instance, portable pH meters. I mean, recently I have bought from Amazon a pH meter that was costing 10 euros, and it worked perfectly just to monitor at home when you're running fermentations. I mean, it has become quite common to have sauerkrauts or to have vegetable fermentations at home, because we want to expand a little bit also the taste that we are exposed to. But again, let's not forget about safety, because this is a prerequisite when we're talking about food. Thank you very much.
Thank you very much, Luca, as well. Very important words here, I think, both in terms of separation between fermented foods and probiotics, because, yeah, we also are very much aware that these are often used interchangeably, to be, and then also, of course, in terms of food safety and thinking about it when you ferment foods at home. Moving on to our final speaker, we have Davide Bulgarelli with us. Davide is an academic who studies the interactions between plants and their microbial communities that help them with stress and grow better. Davide, born and raised in Italy, he now leads a research team at the University of Dundee in Scotland. His work looks at how we can use plant microbiomes to make agriculture more sustainable and climate resistant, and how and now Davide will into the world of soil and roots to show how the plant microbiome impacts the health of our environment and our food system. Davide, I'm very excited for this. Please go ahead, share your screen, and the stage is yours.
And unmute your microphone.
Okay. So can you see my screen?
Yes, you just need to put it exactly. Perfect.
Okay, good. So let me start the timer so I won't go out of time. So thank you very much, Stephan, for this very kind introduction. And thank you very much, UFIC team, to put together this very interesting panel discussion and giving the opportunity to share with such a, let's say, competent and diverse audience what we have been doing in my lab. So let me start with a couple of disclaimers. So the first one, you won't see much of primary data in my presentation. I rather decided to have a sort of graphical summary of what were our main conclusion and main experiments to facilitate and streamline the discussion and accessibility to a wider audience. So if you're interested in the nitty and gritty of what we've been doing and the primary data, we'll have plenty of time in the Q&A or by email to address those. So we have discussed or we have learned a lot about dietary requirements or dietary impact on the gut microbiota. So let me remind you where diet begins. So crops in soil is what basically constitute the vast majority of what we directly or indirectly eat. So therefore, it's important safeguarding those production and make sure that they are sustainable for the future generations. And because soil is one of the most diverse and complex microbial ecosystem on the planet, makes sense investigating how plants may interact with microbes in soil. And what, let's say, has always represented a fascinating element of this type of interaction is the thin layer of soil surrounding plant roots that scientists define as a rhizosphere. So the microbiota, the microbes that are there, are actually quite distinct from the communities that may be characterized or identified in the so-called bulk or unplanted soil, so where is no plant. And at the same time, this interface represents a site of resource exchanges. So plants are pushing in soil organic compounds that have been produced via photosynthesis, and this is to fuel the microbial communities that are there. And microbes that are present there can help plants or actually interfering with some of the processes that plants are supposed to do in soil, such as the acquisition of mineral nutrients. So I hope that this picture, and I hope you agree with me, that this picture looks a lot like, at least operational, to what's going on in our own digestive tract between the microbial communities that are colonizing it and are helping or interfering with our own health. And similar to what's going on in our own digestive tract, it's fair to say that plants, they have their own probiotics, and I put quotation marks here because the definition that we have seen in the previous presentation are quite important. So historically, those are being defined in many ways, such as the most common probably is plant growth promoting microorganisms. And effectively, those the little farmers helpers are taking these organic compounds from the plants and providing plants with ecological services, such as facilitating the acquisition of mineral nutrients from soil, fending off the bad guys of the microbiota, the one that would take from the plants and give back nothing, so-called pathogens, and also helping plant in withstanding environmental fluctuation. That is very critical for the adaptation to climatic modification. So all these things that nowadays, especially in industrialized agriculture, farmers tend to delegate to non-renewable inputs. So then I hope you agree with me that if we get to know a bit better this type of interaction, we may well be in the position to rewire them for the benefit of agriculture. And this has been the overarching question of my research. So the very first thing you've got to do is try to understand and try to know what kind of microbes are populating this interface. And one of the, let's say, the most common ways this has been done these days is by characterizing their nucleic acids, so the DNA. So this graphical representation would illustrate a series of specimens taken from soil or from plants, where the different colors may represent different microorganisms present in different combinations, depending on the characteristics. If you are into this field, so those names mean something. If you are not, if you are a newcomer, just let's say, go with the fact that different colors mean different things, same colors, the same thing. And the way this can be characterized is, as said, by investigating their DNA and sequencing their DNA and basically recreating silico, a sort of digital twin of what happens in soil. And using quantitative approaches, it is possible to characterize or identify what maybe a particular soil does for the microbiota or what a particular plants require in terms of microbiota composition. And this, let's say, underpinning concept was at the very core of an European project that just completed called Circles, that seen University of Dundee participating alongside many others partners in the academic sector and industrial sector in Europe, a project coordinated by the University of Bologna, and UFIC was one of the partners, where we were looking at the food chain microbiota across food chains. And our task was looking at the microbiota of the plant food chain across crops. And here I show you one of the crops we studied, that is spinach, and basically the hallmark of this project, that was a real farm investigation. So we went into the field, into a production site, and tried to understand what kind of microbiota was there with the approaches that I showed you before. And probably one of the most fascinating results we have obtained is depicted in these slides. And again, recall what I told you before, different colors and different combination of colors means different microbiota, same color, same combination of color, same microbiota. So what we have learned is that even under field conditions, so the exposure of environmental, let's say, fluctuation or soil fluctuation, allowed us to identify what are the signature of the plant in the microbiota. So essentially, there is a distinction between the plant microbiota and what will be the unplanted soil. There is a sort of selection. It's not a passive flow through, so spinach wants a certain type of microorganism around them. And despite a general degree of conservation, we were able also to separate between farms. So essentially, we were able to predict what kind of spinach, depending on the microbiota that we were able to identify. But a portion of it was appeared to be conserved across farms. So this motivated us to essentially elaborate a scenario whereby if we are supercharging those microorganisms, potentially beneficial one for the plant, probably we are putting the plants in a better position to withstand what will be environmental fluctuation. Recall, this is an agricultural process. What you saw at the beginning, you have no, prediction of what could happen during the cultivation. That could be flood, that could be drought, that could be potential pathogens. So you need really to prepare ahead of the events to withstand them. And this essentially was our overarching hypothesis. The way you do that in agricultural setting is conceptually similar to what you would do in a clinical setting. So you need to get the members of the microbiota that we have defined modulators in this project, essentially using microbiological media or by mining existing microbial collection, and make sure that the preparation is compatible with the agricultural process. Because, as said, this was a real farm investigation. Anything that differentiates too much from what farmers already do will represent an increased cost. And therefore, you need to make sure that you are able to offset this cost in terms of production. And after that, we were able to monitor the composition of the microbiota. And what has actually struck us as the most distinct element or most distinct characteristic of the treated plants, so the one that received the potential probiotics from the so-called controls that basically were mock inoculated, wasn't not the presence of additional members or the abundance of additional members, but rather some members of the microbiota that were progressively disappearing. And then when we were looking into what they were and what they could do for the plants, we discovered that those may represent or were phylogenetically related to so-called potential pathogen capable of causing yield losses in the plants. And the concept of this biosis that I illustrated in the title of these slides really refer to the fact of individual members of the microbiota are capable of taking over, creating a so-called pathobiome, and therefore creating then yield losses for farmers. So this application was essentially, we anticipated that this application would have been able to reduce this event and therefore represent a significant benefit for agriculture. So this was a snapshot. So where we want to go with that, obviously this is just one crop and a series of farms. But if you think broader and you keep the parallelism existing between the plant microbiota and the medical microbiota, so we may enter the concept of personalized agriculture, whereby depending on the type of soil, depending on the type of crop, you may devise microbiota intervention that progressively decouple profitable production from non-renewable inputs in the agroecosystem. And this is something that goes beyond crop production per se, because microbes are very metabolically very flexible and can really fulfill some of the requirements that have been listed within the concept of decarbonization of agricultural process. And this is where we want to go, in the years to come. This was an enormous effort of several institutions, as I mentioned, across Europe, and I cite here the one that directly collaborated with us in the working package that was related to the plant food chain microbiota. And with that, I would like to close by thanking you for your attention. I'll be very happy to take any questions.
Thanks so much, Davide. That was very interesting to see and to learn about how the microbiome can impact the growth of plants and the yield of the crop eventually. And also the term on the personalization, which of course is also something that is said quite often in relation with the human microbiome and personalized diet, and how that could potentially come to like personalized soil. Also very interesting. Thanks so much. We are at the end of this first section, let's say, and we have still some time for some question and answers. So for this, I'll hand over to my colleague Giacomo. Giacomo, please.
Yes. Thank you, Stephan. So thank you, everybody. We received a lot of questions, both here in the chat, but also in the subscription phase to this workshop. So we try to group them because we received a lot. So we try to group them to answer at least one question for speakers. So maybe we can start from Céline, and please to every speaker to limit the reply in 2 minutes, so we have the time to have one question for each subject. So the question for you, Céline, maybe can be summarized in how we are close to integrate the microbiome science in the medical care, and then what are the applications that can be done in the medical care with microbiome, like to treat obesity or neurodivergence, somebody asked. So
yeah,
please.
So thanks for the question. I think we are now using microbiome in clinical care. So, for example, for patients suffering from Clostridium difficile infection, so an infection that usually patients get after antibiotic treatment, the international recommendation, if the patient does not respond to antibiotic, is already what we call a fecal microbia transplantation. So really the transfer of microbiome samples from a healthy donor to the patient in order to I would say reset the global gut microbiome ecosystem. So it's clearly an application that is used, and we have also more and more therapies that are currently under development. Some in phase 3, so that means very close to the approval. So we hope to see more and more integration of the microbiome in clinical practice. And on the other side, we can also see that it's more and more a concern about in the drug development to integrate the effect of, I would say, classical drugs on the microbiome. So for example, for the antibiotics development, scientists and also the pharmaceutical industry now are trying to develop really targeted antibiotic to preserve the microbiome.
Okay, so apparently we are already making some application, and it's very active science. For Luca, we have one of the most recurring question through the subscription, which is the relation between human microbiomes and impact on mental health. So is human microbiome affecting our mood, and can we change it or influence somehow with our diet? And how long does it take, for example, if this is the case, for microbiome response to the change of diet?
So I'm actually going to be answering from the last question, and then I'm going to go a little bit on the gut-brain axis. So gut microbiome is actually responding quite fast to any perturbation that happens either through the consumption of foods, antibiotics, environmental factors, stress, and all these kind of things. So microbiome is actually, let's say, an acquired organ, I would say, that is really aligning quite well to anything that is happening to us. So, for instance, there are studies where at the beginning of this fascinating field of research have changed, for instance, the diet of animal models from animal-based proteins to plant-based proteins, and you see that the difference in the modulation and the difference in the collagen function happens in matters of hours. So by this point of view, is quite fast as a responding organ. If we take into consideration the new frontier of the gut-brain axis, I mean, it has already been demonstrated how microorganisms in our gut are actually interacting indirectly with our central nervous system through metabolic messengers. Microbes, as I said in my presentation, are really metabolic factories. They are producing a lot of small compounds that are passing the epithelium. They are getting into the bloodstream, and then from the bloodstream, they can actually reach the organs that are very far. And let's say that probably the gut-brain axis is the first one that has been established, but there are a lot of other axes that are actually becoming quite important. So in the future, I'm expecting to have even more axes that are starting from the gut and they go into different organs. The thing that I would like to underline here is that, especially if we focus for a second on the gut-brain axis, we have always gave very important, let's say, or we put a lot of focus on the metabolic exchange, forgetting a little bit that the gut is also the site where most of the neuron receptors are present. So we also have to take into consideration in the past that there may be direct interaction between or mediated interaction between the microorganisms and the neurons that we have, the peripheral neurons that we have inside of our gut. And as a consequence, we should expect that this kind of regulation, and I totally agree that microbes are fully involved in the change of our mood. So based on what we are subjected to somehow, and diet is one of the aspects, but other important elements can be taken into consideration, may perturbate the gut microbiota and microbiome, and as a consequence, we can have also an impact on our mood or worst on our mental health. But again, let's not forget about these two pathways that are the metabolically related actions, but also the contact related evidences.
Thank you. Thank you, Luca, for this complete and very good answer. Last question for Davide. I love the question, how can I improve microbiome composition? How can you use microbiome to improve my orchard? But maybe we have also the question on how in a more, let's say, systemic way, how can plant and soil microbiomes help us in the sustainability challenges, especially if we talk about climate change, and if this can create also an impact in low and middle income countries. So, Davide, please.
Okay, so the question of the orchard, I think we won't be able to answer in a minute. So probably I will need a bit more details on what is not going on that particular orchard. But yeah, in general, this is the answer I want to say. There is no one size fits all type of fix. So it really depends on the problematics of a given environment. Regarding to say, developing countries and the potential for the use of the plant microbiota. And obviously, there is a strong potential because many of these potential probiotic are endemic to a given environment or a given soil. And it's really a matter of identifying what are the trade-offs. So why they are not populating to a point that are promoting the growth of the plant. And this can be done nowadays in a very accessible way, or there are ways coming to the, let's say, coming in terms of utilization that can be applied also in this type of agriculture, where farmers and local communities may retain the ownership of what is the real benefit represented by the microbes. So certainly I would say there is a scope for that, and I do see international cooperation play a critical role on that in terms of enabling the acquisition of this technology, but having this technology on site. But yeah, and then it really depends on the specificity of a particular production system or a particular environment. Drought would require a certain set of microbes that maybe the presence of potential parasitic plants don't. So it's really a matter of defining what are the main constraints and act accordingly.
Thank you. Thank you, Davide. Just wondering if we have enough time for the last question, Stephan, for speakers. Or...
Yeah, I think we can maybe ask everyone to answer in a few bullet points.
Yes.
Let's see.
So we would like to ask every speaker what, in your opinion, as for example, in the perspective of early career scientists, what could be the biggest unknown about microbiomes that we are close to discover, and what does the future of research of microbiome looks like in the future? Who wants to start?
I mean, I can break the ice between the speakers, because I think that what has been happening in the microbiome science is a little bit a kind of amplification of what has been happening previously when the first molecular methods came out from, the development, or let's say the invention. I mean, I remember in the 80s the PCR, and then this blooming application in every single field of research. What has been happening with the microbiome science is that I think that microbiome science probably was mostly driven by technological development than science-based questions. So since we had, after the first years of the 2000s, this huge development in terms of technology, new sequencing machines, very powerful, every single research group that had access to these machines started to do sequencing without really stopping for a second and understanding what was the meaning of this investigation. I mean, as Céline said very, very well, we have a lot of observations, and still we have a lot of observations, very hard to go into correlations. So I think that we need to still move our research from, again, observations to correlations. Probably we need a little bit more to go into function, because we really need to understand what our microorganisms are really doing, how they are interacting between each other with the host. And this is, of course, very, very important for all the things that have been said today. So again, I think that we have, as a scientist, we have been a little bit overwhelmed from the technology. And now we have this huge amount of data. Artificial intelligence is there. This is an opportunity. Let's be careful about it. I'm always a little bit, when we're talking about artificial intelligence, it is, again, humans that have to teach the artificial intelligence. But again, probably now is the moment that we need to really look back what has been done and plan what is best for the future.
if I can complete, I fully agree with what Luca said. It's so true, and I think we will move. Microbiome science will, or maybe should move more and more from composition to function, because what matters at the end is really the function of the ecosystem. Even if the ecosystem is a little bit different in composition, if they are still doing the same function, then maybe the change in the composition is not a big issue. And it's really what we can see also in scientific publication and scientific conferences. There are more and more studies about the function of the ecosystem and less focus on the composition.
Thank you, Céline. I don't know, Davide, if you want to.
But beside echoing what Céline and Luca already told you, and basically second what they've said, I'm fascinated by, in terms of technological development, of what is as becoming the so-called spatial resolution of microbiota science. So right now we are talking in plants and human, most of us are talking about ecosystem, and then basically what we do is wrap together, everything is there, disregarding where they are spatially distributed, maybe sequence them or processing in a different way, and infer readouts. I think this was the logical step one, because the technology allowed us to do so. But now we are in the position that we can really precisely identify where are located certain members of the community, what they can do. And this will provide us with a more nuanced resolution of host microbiota interaction. And probably this will open up for discoveries that at the moment we even ignore. And in terms of translational application will be certainly transformative, both in human or animal and plants.
Thank you, David. Stephan, you want to have the last words to close?
Yeah, thank you. I can have the last word. Well, what else is left for me to thank the speakers? I thought it was very interesting to listen to all of your views on the different areas of microbiome, and especially these last reflective tones. I thought like very interesting and hopefully also somewhat inspiring for maybe the current next generation of microbiome researchers. And yeah, I hope that they also found this event useful. I hope that you had a lot of fun watching this event. Yeah, I think we can close it on this note. Thank you again to all the speakers. Thank you for all the attendees. I wish you a lovely rest of this World Microbiome Day and hopefully a wonderful weekend ahead.
Thank you very much.
Bye-bye, everyone.
Thank you,
Bye.
Stephan. Bye-bye.
Bye. Bye-bye.